Hello, I hope you are doing well. We continue our EpiMed course today, and in this episode I would like to show you some tools to manage omics data from well-known public repositories, for example from NCBI GEO, but these tools can be also applied to some other repositories. Nowadays, we have a huge amount of omics data which are available online, and below I listed some of very popular repositories. Of course, NCBI GEO is probably the top one of omics repositories, which contains different kinds of omics data, for example, in cancer, for normal samples, for different biological experiments. You may also know REExpress repository, which contains essentially transcriptomic data recorded with microarrays. TCGA is a repository for different omics data in cancers. CPTAC is a portal to download proteomics data for cancers also, and GTEx is a huge database for normal samples, normal tissues associated with the corresponding omics data. So today I would like to propose you to choose one example from NCBI GEO. I selected a study in leukemia, different kinds of leukemia. This dataset contains more than 2,000 samples in transcriptomic data obtained with microarrays. And our goal is to see how can we access this dataset, how can we download it, and to pre-process it before we can really start to analyze data. First, we need to prepare the data, and then we can be able to analyze it. Usually, when we work with omics data, we need to manage at least two kinds of data. Of course, the raw omics data. In this data set, we have it's a transcriptomic data set. So we have expression levels for genes in the original format, which are cell files for this data set. And also we need to associate to raw data some biological and clinical annotation for all the samples. In this example, we don't have too much information about the samples, but we have still the sample type, which can be bone marrow or blood, and also the type of leukemia, which can be here B-cell leukemia, T-cell leukemia, acute myeloid leukemia, and other kind of this pathology. So let's together first go on NCBI GEO and to see how this dataset is presented originally on this repository. Let's go online. We are now on the web page of NCBI GEO for this particular dataset, and usually you can find a short description of the study, and also some references to publications related to this study. And I highly recommend you to check this publication essentially for supplementary materials, because sometimes in supplementary materials we can find more detailed description of the samples. Here we have a list of samples, the first 3 samples here, and at the end of the page we can find a download link to download manually, if you want, to download raw data of the entire data set. Just be careful, because the size of the file can be very important. It can take time. In EpiMed, we never download the data manually. We use always a special tool developed by Floran. So this tool is a R package, which is named EpiMed Tools, and this package is completely open source. You can download it from its GitHub repository. You have a link here. So this I invite you to go to this link and to explore the package. You have a documentation. Normally you have a documentation which explains you how to install the package, and also in Vignette's directory you will have some example how to use it. So this package allows you to properly download the data, also to pre-process the data from its original raw format to more readable, human-readable format, then to normalize data, and also log transform the data. So you will be able to perform all the steps in your pipeline before you can start the analyzing of data. Now let's see how can we work with bioclinical annotation of samples. This annotation can also be problematic for data processing. Let's look first how it is presented on the website. From the page of the data set, we can see that there is a list of samples listed here. Let's look to the first sample just to see what kind of clinical information is available for this sample. So here we don't have too much information, but we can see that this sample, this particular sample, is annotated with just with some text, non-structured text. It's explained that it's a leukemia, and the sample type is bone marrow, and the leukemia class is B-cell leukemia with a particular translocation, so a particular cytogenetic type of this leukemia. And that's almost all. We know also that the patients were not treated before the sampling. Of course, they were treated after the sampling, but we can say here that our data are not impacted by the treatment, chemotherapy or radiotherapy or something like this. And then we have some technical information about how the sample were processed before measurement. But it's not, here we don't have too much clinical information. And that's almost all. At the end of the page, we can sometimes have a link to the data, to raw data for this particular sample. The problem with the clinical information on NCBI GEO is that the clinical information is usually presented just as a text or plain text in a non-structured format. And the unstructured text format is difficult to use directly for analysis. So first we need usually to convert the plain text into a more structured format or completely structured format to be able to analyze this clinical information later. And as you can see in our example, the clinical information is just a text, and we need to structure it and sometimes to deduce the information which is not present in the text. So it would be nice to represent the same information in a different way. For example, to create some categories, for example, category tissue, and to give a value to this category. In this case, it's a bone marrow. And also when we talk about tissues, it's important probably to classify all the possible tissues within a standard classification. It exists already different international classification, for example, the International Classification of Disease for Oncology, and inside this classification, the bone marrow is just codified like this. So it would be nice to recognize automatically that the bone marrow here in text is in fact correspond to the identification number of the tissue in this particular classification. We can also be interested to introduce probably a category pathology and to say that this sample is a tumoral sample, it's a cancer. Of course, we can deduce it, a human brain can deduce it very easily that it's a cancer because we know that it's a leukemia, but the computer doesn't know that a leukemia is a cancer. So we need somehow to guess or to deduce this information, because if you have many samples, it would be nice, for example, to to select among all the samples only tumoral samples for data analysis, or only normal samples, or samples with a particular pathology. So it would be interesting to have this this category. Also, we need, of course, a subtype which describes exactly what is a particular type of this cancer. So it's just an idea presented in a structural format, which is a JSON standard format, but we can also represent it in Excel-like format with columns, and we have the information in each column. It would be nice to have a standard number of columns, always the same. In this case, we can consider different samples and to compare samples between them, and compare different data sets between them. EpiMed database can help you to organize clinical annotation from NCBI GEO, for example, and if possible, it can also to automatically recognize unstructured text and present it in a more structured format. It's not always possible, but sometimes it can be done. And then if you use EpiMed database to do this, you will be able to download all the clinical data in an Excel format or CSV format. So first we need to go to EpiMed database and to import a study from an external database, NCBI GEO. In our example, we need just to put the accession number of the study we are interested in. It will be imported in EpiMed database. It will be recognized automatically, and then you will be able to download this study in Excel format here. So let's see how can we do it. Yeah, so first we should go to EpiMed database, and the first step is to import a study from an external database. So here we decided to use our example. We put the accession number of the dataset, and we submit it to EpiMed database. So it happened that this particular study already exists in EpiMed database, and it's already imported and recognized. But if it's not present, then on this page you will see the process of importing of the dataset in EpiMed database, sample by sample, automatically. Then when it's finished, then you should go to clinical data, download experimental grouping. Experimental grouping means the same that just bioclinical data. Then you put the accession number that you want, and you can be able to download the clinical information in such a way in Excel. Let's look inside the downloaded file. First, you can notice that we have two tabs. One tab for original parameters and one tab for standard experimental grouping. Let's first look at original parameters. The original parameters are in fact exactly the same parameters, exactly the same information that we can find on NCBI GEO website. Just download it here. Download it in and also present it in columns, presented in a little bit more structured way. If you would like to compare different data sets between them and compare different samples, it's not sufficient, and we need sometimes to recognize these original parameters and to project them to well-known, established and standard columns that we prefixed for all the data sets. So let's look at these columns. First we have the identification number of the sample, and the identification number of the study, the identification number of the platform, which is it means microarrays here. Then we have sample title as it presented on NCBI, and sample source also is presented on NCBI, the first columns. And then we try to recognize the original parameters, the original text, in terms of some categories we are interested in for all the for all the data sets. For example, we would like to know if the sex is marked or not for patients. So in this case, we don't have this information, so it's empty. The column is empty, but when the information is present, if it's in a non-structured way, here we will have the information in a structured way. Ethnic group, age, so we don't have this information, but it could be recognized if we have the information in the original parameters. Then we have the tissue stage. We know that this sample contains adult cells, adult tissues, and sometimes we may have, for example, fetal tissues or embryonic tissues. This information is not directly available in original parameters. This information is deduced by the tool, is deduced from original parameters and from the global description of the study. We know also that these samples are tumoral, which is not, this information is not directly present in original parameters, also deduced from it. And the pathology is cancer. We have also the information that is bone marrow, but the word bone marrow is recognized from the international classification of disease for oncology, and we have an identification number for the tissue in this classification, and also some other parameters. For example, we have also the identification number for leukemia, for this particular leukemia, which is B-cell leukemia. And finally, we have the subtype of the leukemia, which is B-cell with a special translocation of leukemia. Sometimes in the original parameters, we may have the information about T and M stage. So in this case, we will have the information here, and we are always interested in data set which have the information of survival for patients, disease-free survival or overall survival, or the both, if the patient is relapsed or not, and if the patient is dead or not. Finally, we have also another classification of tissues, depending on level. Sometimes it may be helpful. So that's all. In the clinical data, we have usually two kind of information: the original parameters, it's presented on the website, and standard experimental grouping, which is deduced from original parameters. And this is not the original information; it's deduced and pre-processed, just to simplify the analyzing of data. Well, I think that's all that we have. Yeah. To summarize, I leave here some links, a link to our package EpiMed tools for raw omics data processing, and also a link to EpiMed database for clinical data processing. If you have any question, please send us an email to the mailing list. Thank you for watching. I wish you a good day.